So what do we mean specifically by gene expression? This is when we actually build a protein from a specific gene. So the gene in the DNA is there all the time. It's being expressed, it's being actually used when we do build a specific protein. The basic steps of gene expression, we're going to go from DNA to messenger RNA to a protein. The first step is called transcription. And transcription is when you copy a small part of DNA, which is a gene, and create a piece of messenger RNA from that. And we abbreviate messenger RNA as mRNA. Then you take the messenger RNA and you go through a process called translation. And translation is when a ribosome reads the messenger RNA and uses that to build a string of amino acids. And of course, a string of amino acids will fold into a completed protein. So two key parts, transcription and translation. So a little bit of review about DNA and RNA. If you look back at the work we did in Chapter 2 and biomolecules, we talked about a lot of this already. So it's one of the 4 macromolecules needed to build a cell. Its monomer is a nucleotide. A nucleotide has 3 parts, a sugar ring, a phosphate ring, and a nitrogen base. DNA is going to be a double-stranded molecule. RNA is single-stranded. DNA is found only in the nucleus of a cell. RNA, there's different kinds, but they can be found in the nucleus and in the cytoplasm of the cell. DNA is going to be created by a molecule called DNA polymerase, and DNA is going to be copied once in the life cycle of the cell. RNA is created by RNA polymerase, and it's going to be made millions and millions of times within the cell cycle. In DNA, we have our base pairing rules. Adenine binds with thymine. In RNA, adenine will pair up with uracil. I mentioned that there's a couple different kinds of RNA. There is messenger RNA, ribosomal RNA, and transfer RNA. There's actually a number of additional kinds of RNAs, and this is a huge area of research in biology right now to understand the roles of these additional RNAs. But these are the basic ones that we need for gene expression. So messenger RNA, or mRNA, is going to take the message from the nucleus to the cytoplasm. Since the DNA does not leave, messenger RNA is made during transcription, and it carries a transcript of the gene to the ribosome. Ribosomal RNA is used with other proteins to build ribosomes. So the ribosome itself is a complex including rRNA and proteins. The ribosome, of course, is the organelle that actually builds proteins. Transfer RNA is floating around in the cytoplasm, and transfer RNAs bring single amino acids to the ribosome. Each type of amino acid is associated with a unique tag, specifically called an anticodon, on the transfer RNA. So the transfer RNA is this complex 3-dimensional molecule. On one end, it's holding on to an amino acid, and on the other end, it's got an anticodon that indicates what kind of amino acid it's carrying. So I use various goofy examples in this class, and you could think a little bit about gene expression as a restaurant actually making a recipe. So the cell is the restaurant, and we've got the main kitchen, and then you've also got the office for the chef where he or she has their master cookbook. So think about DNA as the master cookbook for the restaurant. And every day the chef is going to pick specific recipes that he or she is going to make for that particular day. That master cookbook is pretty valuable, and you don't want to take it out in the kitchen and get flour or oil all over it. So instead you make a photocopy of just the pages of the recipes you're going to use. So let's say today we're only going to make apple pies and cheesecakes and nothing else. We're going to photocopy the apple pie recipe and the cheesecake recipe, and we're going to send that out into the kitchen. We're going to hand it to our chefs, and the chefs are going to make that recipe. That's what's happening with gene expression. We're taking a recipe and actually making it. And if we look at the different kinds of RNA, they're going to support this process. So if DNA is the master cookbook, messenger RNA is the copy of that recipe. Ribosomal RNA and the ribosomes are going to be the chefs. They're actually implementing what the recipe says. And the transfer RNAs are the sous chefs. Sous chefs in a kitchen are going to prepare ingredients and bring them to the chefs. In this case, you have a different sous chef for every single ingredient. So someone's bringing you eggs, and someone's bringing apples, and someone's bringing sugar. Eventually you put all this together and you end up with a apple pie, or in this analogy that would be the final protein. So I want to show you in a little more detail how the messenger RNA and the tRNAs might work together, the transfer RNAs. A codon is a three-letter code on the messenger RNA. So here we have a piece of messenger RNA, and it's got this long sugar phosphate with different nitrogen bases hanging off. It's single-stranded and it has U's in it, so we know that it's RNA. And a codon, again, is just three of these nitrogen bases. An anticodon is a three-letter code on the transfer RNA. So here we have a transfer RNA. At one end it's got the anticodon, and at the other end it's holding on to an amino acid. This one's carrying alanine, this one's carrying leucine. And notice that each one has a unique order in its anticodon. So the codons and anticodons can match up by base pairing rules. The codon and anticodons are not identical. Instead, they're going to use base pairing rules. So C is going to bond with G, U bonds with A, and A bonds with U. And so this codon matches up with this anticodon. That means that this code is asking for a leucine next. Then we can move down to the next codon, which would be GCG. And if you think about what will match up with that, what matches with G is a C, C would be a G, and G would be a C. And notice CGC, that would bond with this next codon, anticodon. And so the code is saying the next thing we need is an alanine. So our messenger RNA codon and our transfer RNA anticodon are matching up by base pairing rules.